Alright, this is the 331 review, and you covered two things. You covered about cellular division for mitosis and meiosis, and you covered genotypes and phenotypes. So let's talk first about um, mitosis. So mitosis is that's happening all over your body, all over the time, all the time, and it's basically where one cell, and you have all your 46 chromosomes, copies itself into two cells. They both have the same amount of chromosomes. All right, so they make identical cells so they can go and do the same thing. So if your skin cells die and they're replaced almost like once a week, that's where all dust comes from all the skin cells coming off of you. And mitosis just constantly replaces them. Otherwise, we would have no skin. So that's mitosis. Meiosis is a little bit more complicated. So what happens is when you create a baby, the sperm and the egg come together to make an offspring. All right, so you get half of your chromosomes from your dad, or half of your genes, and you get half from your mom. Well, since we get half and half when these two join together to make the baby, the offspring, you can't have 46 plus 46 because then you would get 92. And that's way too many chromosomes. In fact, having just one extra is bad for the baby. And that's Down syndrome. So we have the dad might have 23, and the mom has 23, and so we can get a baby of 46. So in order to do this, we have to go through meiosis. So meiosis is you have one cell with 46 chromosomes, and it creates four cells that all have 23. All right, so now the other thing that's important is that these are all different. All right, so if we talk about um, our chromosomes, so mitosis is the same. It just makes copies of the chromosomes and then divides them between two cells. Meiosis does the same thing, but divides them about four cells. And so if we take, let's just take a cell with four chromosomes. So 1, 2, 3, 4. All right, so these chromosomes are paired. And what happens is when it divides, I'm not going to show all the steps, but I'm just going to show the final step with the four chromosomes. The four cells. Is in one cell, the red and green might go to one cell. In another cell, the black and green might go there. So you get the black chromosome and the green chromosome. Another cell, you might get the red chromosome and the blue chromosome. And another cell, you might get the blue chromosome and the black chromosome. So these give several, and these have different genes on them. So therefore, you have a chance to make four different babies. So if you're talking about, now this is only with four chromosomes, if you're talking about 46, each time this goes through, you're going to have a different variety each time you go through meiosis. So that's why all the brothers and sisters are always different, because there's so much variety of how these guys can randomly get sorted. All right, so that's meiosis, and meiosis creates those variety of cells, so they're all different. Now, what you guys did is worried about inheritance. So you guys exchanged chromosomes, and you found out that if this was the guy here, if this was the male, these would be the sperm, And then this would come together maybe with a female egg over here. Only one of these lucky guys get to make the baby. And then you end up with a cell. Let's say this one has let's say this one has a red one and a green and blue one. And then we end up with a baby that has blue, 2 reds, and a green. So not only is the baby is somewhat similar, so like the dad, it had similar right here, this similar combination, but with the mom, but with the dad, it has red and black. This one has red and red. All right, so they're not going to be exactly the same. And so, example, and I didn't show the starting cell. Let's say the mom's genotype is the same is red and black and blue. So instead of being like the dad where it has a blue and a green, they're both similar and they have red and black, but then she has blue and blue, he has green and blue. So the chromosomes, all 3 are different, and this is just again with 4 chromosomes. Imagine if you have 46. All right, so these create a whole wide variety of different offspring. So the next part then is, before we get into the next thing, you guys have to understand all the different vocabulary terms geneticists use to describe sexual reproduction and the chance of getting different genes and such. So we have these different butterflies here. The genotype are the genes. So if I would have, on the last cells, instead of making green chromosomes and red chromosomes, if I would have put letters on them, genes are usually shown by letters like an A and maybe a little a. That would be the genotype. So those are the genes that the people are getting. All right, so there's 3 different possible genotypes if we're only talking about one gene. You can have 2 dominant. All right, so dominant is the capital letter. You can have 2 recessive down here. Recessive is the little letter, or you can have a dominant and recessive. All right, and that is one of each. These genotypes then determine our phenotypes, and the phenotype is just the trait. All right, so you can see here is that both of these are brown and this one's white. So those are 2 different phenotypes. Now, the reason why this one is light is because it only has the recessive letters. When only the recessive letters are present, then you get the recessive phenotype. All right, but you have to have 2 little letters in order to get the recessive phenotype, because when you have one of the dominant letters present, it is going to be dominant, which is the dark phenotype in this situation. All right, and then this here has a special name. Big A, little a is called a carrier. The reason why it's called a carrier is because, especially if we talk about a genetic disease like sickle cell, this person doesn't have sickle cell because the dominant overrides recessive, but because they carry the recessive gene, meaning they can pass it on. All right, so we call people who are capital lowercase, we call them carriers because they carry the recessive gene. All right, so those are the most important terms. I mean, there's homozygous and heterozygous. These 2 would be homozygous because they're the same, and then this one's heterozygous because it's different. But that's not a term we're focusing on in this class. But you definitely need to know carrier, which is the heterozygous genotype. All right, so now applying this to sickle cell and the chromosomes that you did. So you did Best disease, sickle cell, and hemophilia. And they all had slightly different characteristics. Best disease was dominant, hemophilia was sex-linked, and sickle cell was recessive. So if you did Best disease, just to review this, Best disease, you would have had Best if you had BB or BB, big B, little b, because Best was dominant. Whereas sickle cell is recessive, so you would have to have little b, or little s, whatever letters you want to use, it has to have two little letters in order to have sickle cell. All right, you would be normal if you had big B, big B, big B, little b. These are normal. Whereas in this instance, little b is the normal. All right, so that's the difference between dominant and recessive. And then hemophilia was sex-linked, so what that meant is we have the X chromosome and we have the Y chromosome. The X chromosome is only found in females. Not only. If you have two X's, you have females. If you have an X and a Y, you have a male. Well, the hemophilia gene was only on the X chromosome. So it was recessive, just like sickle cell, but is only on the X chromosome. So if the male could either have big H or just little h. So they don't have that chance to be heterozygous, or they don't have these chances here, be big and little. They're either big or they're little. So they either have hemophilia or they don't. They're not going to be carriers. Only females can be carriers. All right, but I want you to focus on sickle cell. So sickle cell, they use two different letters in this diagram, which is dumb. Always use the same letters, so I'm going to use big A and little a here. I'm going to change these. All right, so there's sickle cell. So if you have two parents, both of these are carriers because they each carry the gene, but they don't have the gene. They can have different possibilities, and this is where we're going to next. They can have a possibility of having a big A child who is normal, has two normal copies. They can have a chance of having carriers, and they can have a chance of having sickle cell. They can have every possibility because she can donate a little a, and he can donate a little a. She can donate a big A, he can donate a little a. She can donate a little a, he can donate a big A, and then they can both donate big A's. All right, so you just kind of have to look at what they can give to give the simple combinations. And then that will lead us, if you remember, eventually we're going to get to Punnett squares. And then that makes it a lot easier to see what they can give. All right, that is it for this section.